Yes, sir, you can start now.
Thank you, ma'am. Thank you so much. Good evening, my dear students. You all are warm welcome once again. The wonderful session of the biological classes, the chapter number 22, molecular basis of inheritance. Already we studied, which one, before this chapter, the principles of inheritance, isn't it? These two chapters, very, very important. Most of the questions, the number of questions will be very high in this two chapter only. Maximum questions up to 12. Okay, from these two lessons only. So be carefully and study the lesson. Don't left it and don't omit the lessons. Okay. Today in this lesson, what we are going to discuss, the molecular basis of the inheritance. Which molecules responsible for the inheritance process already very about, isn't it? Normally, Ajay, will you please tell me which molecule is responsible for the inheritance process?
So,
Yeah.
so RNA, sir, RNA and
DNA.
DNA. And
Yes, very good. Both of correct, but the major component, which one, Dhanalakshmi, DNA is the correct one. RNA is also supporting, isn't it? So RNA is responsible for the biosynthesis pathway, particularly in the protein biosynthesis. Okay. So the molecules, which one is the higher molecular weight compared to these two? DNA is the high molecular weight compared to RNA. Okay. See here. Here, see that what are the questions in our mind, So what is meant by genetic material? How will you prove? For example, so DNA has a genetic material, isn't it? Why? Because it transfers one generation to another generations. Already very about mental laws. Okay, law of dominant, law of segregation, law of independent assignment. So DNA is a major role. It plays a vital role or major role in the segregation process, one generation to another generations. Okay. Okay, genetic material, right? Okay, it has a specific allele and also it carries a specific characteristics. What are the characteristics you are having? The potential effect will be expressed each and every generations. Which one DNA. Okay. So how will you prove DNA as your genetic material? Any ideas, my dear students? Ajay and Dhanalakshmi. How will you prove DNA as your genetic material? How will you prove? Any idea?
no, sir.
No. Okay. What about Dhanalakshmi?
No, sir.
Okay. What about the Griffith experiment? Pneumococcal, isn't it? That is the proof of genetic material, DNA as a genetic material. You know all about that? He conducted the experiment with a mouse or mice, isn't it? Smooth and rough strain. Yes, as well as the denoted as smooth and R denoted as rough. They injected properly directly to the mice. What happened, mice will be, or when it injected, what happened, the mouse is alive. Suppose smooth injector, it dead. Okay, after that, R plus S, what happened? Mice is also heat killed, but that process also we'll discuss later. Okay, then who discovered DNA as a nuclein? The first they have given as a name is called as, what is that? They didn't give the name as a nucleic acid. The first they give a name is called as nuclein. Some of the scientists there. And then nucleic acid, who give the name nucleic acid? This is the one. Who discovered DNA? Already in our report. Okay. There are three major scientists also involved in this process. And who discovered the RNA also? Okay. RNA is nothing but the ribose nucleic acid. DNA as the deoxyribonucleic acid. Both are nucleic acid only. Okay. But the DNA is a high molecular weight compared to the RNA molecules. It's mostly found in where, the nucleus. Okay. This is a very, very important one. Another one is RNA, ribose nucleic acid. Here, there may be a smaller molecular weight. It's also produced by DNA. The process is called as transcription. DNA into DNA, that is called as what is that? Replication. DNA to DNA, replication. DNA to RNA, transcription. With the help of the DNA and RNA, the protein biosynthesis takes place. That condition is called as what is that? Translation. Transcription, replication, translation is also there. Okay, this is very, very important. And the next one is what is meant by Chargaff rules. This is very, very important. It is depends upon the nitrogen bases present in the DNA and RNA. We'll discuss one by one. Okay. And then how many types of bond formation in DNA molecule? How many bonds formation present in it? For example, already you studied. How many bonds is there? Hydrogen bond, covalent bond. Okay. And also disulfide bond, sulfide bond. Okay. And a weak bond is also there, isn't it? But the DNA molecule construction process, what are the bond is also present. This is also very, very important. Glycosidic bond is also there. Peptide bond is also there. Okay, carbon also connected with another carbon atom. Nitrogen is also nitrogen. Nitrogen with the hydrogen. Likewise, so many bond is there. But particularly in this DNA molecule construction, what are the bond is present? And it is also very, very important. Okay. Okay. Go to the next one. See the one. So, what is the correct sequence of the order? What is the correct sequence order of the genome? This is the correct way. Note it down, my dear students. This is very, very important. Suppose take an example. So nucleus inside is the nucleus containing genome first. Note it down, please. Nucleus, it contains genome first, then chromosome, then genes, then nucleotide. In between, there is a alleles. Okay, genome, chromosome, gene, allele, nucleotides. So these are the very, very important. So this is the correct sequence. Okay, don't forget, my dear. This is very, very important. Sometimes they may ask the question, arrangement of the correct sequences like that. Okay, see that. See here. So there is an expansion is there. See that. Identify the correct order of organization of genetic material from largest to smallest one. Largest to smallest one. The correct option is D has given. What is that meaning? The arrangement is genome, chromosomes, gene, nucleotide. This is the correct options. So what is by genome? First of all, you should note it down. What is by genome? A genome is an organism complete set of DNA. Particular animals or any one of the organisms. Take an examples of plant or animals, particularly human being, whether it may be lower or higher animals. Okay. The genome is, what is the meaning of genome? Complete set of DNA, including all of its genes. Take an examples. How many genomes we are having? 3.4 to 10 to the power of 16. Genome is present in our body. But total length of the DNA molecule, you know about each and every human being, there is a DNA nucleotide length only 2 meters only. But the nucleotide will be very high. Okay, see here. genome first. A genome is an organism's complete set of DNA, including all of its genes. Each genome contains all of the information needed to build and maintain that organism. All the genes, they are having the information. What is the meaning of information? Information, nothing but your characters, skin color, diseases, blood group, whatever it may be. Okay. So these are all are the information in the form of which one, nucleotide. Okay. So this is the first one, genome. Okay. Genome followed by chromosome is there. G, C, and G, N. G, C, and G, N. G denoted as genome. Chromosome, again gene, again nucleotide. Chromosomes, chromosome are the thread-like structure located inside the nucleus of animal and plant cells. Chromosomes are the thread-like already in our world, but the structures located inside the nucleus of animal and the plant cells. Already we studied in the mitosis, meiosis. Chromosome look like a thread-like structure. When it condensation takes place, it look like a, what is that? Bivalent nature. That things we will study in the cell division, cell cycle. Okay. Next, genes. A gene is a basic physical and functional unit. Note it down. Already you know about it. Which one is the functional unit of the living organism, cell. Okay. But here, the functional unit is, what is that? Hereditary. The character will be transferred from the one generation to another generation. Okay. That is called as gene. The genes are made up of DNA. Okay. The genes are made up of DNA. The DNA is made up of nucleotide. Okay. So nucleotide means already you know, but here you should very about TIDES, tides. The building block of nucleic acid or the nucleotides. What is by nucleotide? What is by nucleoside? Dhanalakshmi? Do you know idea?
No idea, sir.
What about Ajay? I think you know very well. What is my nucleotide? T. What is my nucleoside? Yeah.
So,
Yeah.
so nucleotide is, so it's basically, so the histone and the DNA stand round, sir, wound around the histone.
Okay. Okay.
Nucleoside is part of DNA except that phosphates, that sugar part, sir.
Okay. You are slightly touched to the matter, but it is not complete. Okay. See here. So nucleotide, which one is the T? T denoted as, you just note it down. T denoted as 3. Okay. 3 means what is it? Each and every DNA molecule constructed by 3 component. One is, suppose if it is DNA, deoxyribose sugar molecule. First one is sugar molecule. Second one is nitrogen bases. It may be purine or pyrimidine. So 2 components came. Third one is called as phosphate. Suppose if it is these 3 components, which one sugar molecule, nitrogen base, and also phosphoric acid. These 3 components present in it. It is called as, what is that? Nucleotide. T. 3 components. Suppose absence of phosphoric acid, it is considered as a, or named as a nucleoside. Yes, second. Okay. You got now? Ajay.
Yes, sir.
Okay. Will you please repeat again? What is my nucleotide?
So it's the building block of the nucleic acid, sir.
Okay, good. What are the components is there? Please take a notes. If it is better for you. Nucleotide. What are the components is there? Please, please. Now only I told you, no? Okay, dear. Listen
Sir,
here. Once.
sir,
Yeah.
So it's basically, so it consists of nucleoside and a phosphate, sir.
Very good. Simple. Nucleoside and phosphate. What is by nucleoside? Nucleoside means it composed of two component. Sugar molecule and another one is called as what is that? Nitrogen
So
base.
nitrogenous base, sir.
Okay,
Nitrogenous base.
don't forget, my dear. This is very, very important. Okay, okay, good. Super. So Dhanalakshmi, please follow me. Okay, see, yeah, that's why I told you. See the next one. So DNA is a acid or base? Another questions. Okay, DNA is built of both acidic and basic component. That is amphiphotic nature. Okay. The acidic component of the DNA is its phosphate group, that is called as phosphoric acid. And the basic component of the DNA is its nitrogenous bases. Don't forget, this is the base, this is the acidic nature. So the DNA is built of the both acidic and the base components. Which one is the base? Nitrogen is a base. There is nothing but the purine and pyrimidine nitrogen bases. Okay. Phosphate, this is nothing but the phosphoric acid. Okay. So it is a acidic nature. So these two are present in the DNA molecule. It is very, very important for the construction and also the DNA molecules. See here. Okay. So now we enter into the lesson. These are the preview or otherwise called what is the intro of the DNA molecules. So first discovery of the DNA, that is nothing but the historical aspect. It is very, very important. Who first discovered the DNA molecule, what they named. So many times they were asked the question, my dear. Take a notes, please, my dear. In 1868, don't forget, John Frederick Meisser. Okay, see, John Frederick Meisser developed for isolating the nucleic from white blood cell present in the pus. You know about the pus cells? Pus cell means after wound, 3 to 4 days, what happened? The white color fluids comes out. That substances was taken by the Frederick and also analyzed. Okay, what they analyzed, how they gave their names, the minute amount of nuclear material that he obtained contained novel substances that was slightly acidic and high in phosphorus. So the Meisser called this material nuclein. Don't forget, my dear. Who gave a name nuclein means the Meisser. Which year? 1868. Which substances they have taken as a test solution? The pus cells, okay, from the wound, okay, which considers a DNA and protein. Okay, sometimes they may ask the questions. The Meisser first isolated the nuclein, which consists of what are the component DNA and protein molecules. Don't forget, my dear. Okay, so what is that meaning? Ajay, please, who first discovered the nuclein?
Sir, Meisser, sir.
Meisser. Very good, very good, good. Okay, so what are the component present in it, my dear Dhanalakshmi? What are the component present in the nuclein? Dhanalakshmi?
So DNA.
DNA and
Protein, sir.
okay, don't forget, my dear. Okay, so these are the very, very important. Sometimes they may ask the question, so many questions from this part. Okay, so many times I'm asking. Okay, what is the next one? See, and Bobus Arnon Liban discovered that DNA consists of large number of linked repeating unit called as nucleotide. Who discovered nucleotides? The name of the scientist called as Bobus Arnon Levine. Okay, nucleotide was first discovered by P.A. Levine. Next, 1953. Don't forget, my dear. This is very, very important. The DNA molecule double stranded helical structure was discovered by the scientist called as, what is that? Watson Crick. So 1953, double helical structure of the DNA was given by James Watson and Francis Crick. Based on their X-ray diffraction, they produced Maverick, Wilkins, and Rosalind Franklin. Don't forget. This is the part very, very important. How the DNA molecules was discovered, with the help of the scientist name is called as what is that? Maurice Wilkinson and Rosalind Franklin based on the X-ray diffractions. Note it down, my dear students. This is a very important question. This part only. Double standard helical structure was discovered by the name of the scientist Watson and Crick. Only we studied it. Full name is James Watson and Francis Crick. How they discovered it? With the help of the X-ray diffraction produced by which Maurice Wilkinson and Rosalind Franklin. Okay, this is the very, very important. The next one is the hallmark of their proposition was base pairing. How they construct with the help of a nucleotide only. The nucleotide is made up of which one? Sugar molecule, base pairing, and also phosphoric acid. Particularly, which one is responsible? Construction in between the two strands, there is a which one, base pairing. Okay, without base pairing, what happened? There is no DNA molecule double standard. Okay, that is called as what is that? Polynucleotide. It is not a one nucleotide. It is more than 1,000 millions of nucleotide. That's why they called as the polynucleotide chains. Okay, see one by one. This is very, very important historical aspect. Okay, particularly who discovered DNA molecule, who discovered nucleotide, double-handed helical structure, who discovered, based on which one. Okay, the X-ray diffraction is there, and hallmark, which one, base pairing. Just to go through it, already I have sent the 5 set of question 2015 onwards. I have sent the original question paper to our website. I mean WhatsApp. Please check it. Which one is very, very important. Just go through it, my dear students. For the next one, this was based on the observation of the Erwin Chargaff. The nucleotide, they based on the observation of the Erwin Chargaff. How? What is my Chargaff observation? Already in our board. See, I see that one. See, my dear students. See, first one is what is that now? DNA, isn't it? DNA. DNA nothing but what is that? Deoxyribonucleic acid. Structure will be like this, isn't it? Like this. Here, what are the component present in it? What are the component present in it? See, there are 3 components. Is there? What is there? First one is sugar molecule. Sugar. What is that? Sugar molecule. First one. First one is sugar molecules. And second one is, what is that? Already told you. Nitrogen base. Okay. And third one is, what is that? Phosphate molecules. These three are present, that condition is called as what is that? Nucleotide. Just imagine, my dear students, nucleotide. Okay. Suppose the phosphoric acid is absent. Okay. That is called as what is that? Nucleoside. Okay. What is that? Nucleoside. Okay. Don't forget. So In between, okay, this is a stand, it's a double helical structure. This was discovered by the name of the scientist called as Watson Crick. Suppose if I draw in a, just like a ladder, okay, just a without coiling, with a twisted structures, I have written, drawn here. In between, what happened, there is a nitrogen base is there in this place. Okay, how, adenine molecules, okay, combine with the thiamine, okay, guanine molecules or nitrogen bases join with the cytosine. Here the double bond, adenine always pair with the thiamine molecules with the help of double bond. Here the 2 is there, isn't it? 2 bond is there. What about in this area, guanine connected with the cytosine with the triple bond. This is very, very important. These are the nitrogen bases. Okay. So the outer side, what happened, there is a sugar molecule is there, and followed by there is a phosphate molecules. See here, sugar is the sugar molecule, the center, and nitrogen base is also there. Here, the nitrogen base is also connected with the 2 strand. So this one is called as 5 dash. This one is called as 3 dash. Here 5 dash, here you have to put 3 dash. Here 3 dash, here opposite there is a anti-parallel each other like that. So what are the things, see, these are 3 things is very, very important. These are the 3 things is there. See that nitrogen base is there. This one is called as nitrogen base. This one nitrogen base comes here. Sugar molecules also there. Phosphates. This is called as what is a nucleotide. Okay, this is called as nucleotide. These 3 component present. Same aspect also opposite side. So the nitrogen base is there, and also sugar molecules also there, and addition with the phosphate molecules. There's 2 chains. The 2 chains are connected with the bond is called as the center. There is a 2 bond is there. There is a triple bond is there. You got it now, my dear? Okay. So this is the DNA molecule. So once again, okay, I will show you in the then it will be better for you. Okay. See that? So it can be easy, better for your identification. Yes. See, this is the correct. Okay. See that structure will be. See, this is also confused. Okay. See here. This is the structure of DNA molecule. Okay. See that this is the structure of DNA molecule. It's also confused slightly for you. Okay. Anyhow, this little. Okay. See, media. Okay. See here. So the Chargaff observation was that for a double stranded DNA, the ratio between the nitrogen bases also divided into 2. One is called as purine, another one is called as pyrimidine. Okay. There are 2 basic things is there. Okay. Nitrogen base also divided into 2 types. What is that? Nitrogen bases divided into 2 types. Okay, purine and pyrimidine. Purine is also what is that? Comes under the headings purines. There is adenine guanines also present in it. Pyrimidine, see that one. So pyrimidine, what happened? See here. Cytosine, uracil, and thiamine is also there. Once again, repeat for you. A nitrogen bases divided into 2. Purine and pyrimidine. purine, adenine, and guanine. Pyrimidine, cytosine, thymine, and uracil also there. These are the hydrogen bases present in the DNA as well as RNA. Sugar component. Which one is the sugar component? Deoxyribose sugars and ribose sugars. I will tell you that things also it's better for you. I will come from the previous one. Then only it will be better for your identifications. Okay. See, adenine, guanine. Okay. Thymine and cytosine. Just see the third letter. What is that? Here, thymine. How will you differentiate? Which one is purine? Which one is pyrimidine? How will you identify, my dear? Ajay, how will you differentiate? Which one is purine? Which one is pyrimidine?
So purine.
Purine. What is that? Please go ahead.
Sir, purine will be, sir, it's adenine and guanine, sir.
Very good.
Sir, cytosine and thymine is pyrimidine, sir.
Yes, very good. Okay, good. Very good. See that suppose if you have
Sir,
any,
Sir, this purine has double bond in between, sir. Pyrimidine has triple bond.
So not only that, like that, Chargaff rules also. What is in that? You know that Chargaff rules. One purine molecules always united with the another pyrimidine, or one purine, one pyrimidine. Likewise, so adenine always react with the purine molecules of adenine joined with the pyrimidine molecules of thymine only with the help of the double bond. I will show you. Guanine always is a purine joined with the pyrimidine molecules of cytosine with the triple bond. So both are equal. Number of adenine, how many number of adenine molecule present in it? It is also equal to the thymine molecules. Number of guanine is also equal to the cytosine, directly proportional to the composition or number of cytosine molecule. So both are constant. Adenine, that is equal to thymine, that is equal to 1. Guanine and cytosine is also equal to 1. Okay, don't forget, my dear. Very good. Keep it up, my dear. Okay, see. So first one is nucleic acid. The nucleic acid are polymers of nucleotide. Nucleic acids are polymers of nucleotide, otherwise called as DNA molecule. A nucleotide is a phosphate ester of nucleotide. The otherwise called as what is that? Phosphate ester of nucleotides. Okay. Which one is the nucleotide? See, DNA is a biomolecules which play a very important role in the process of inheritance. Already told you, isn't it? Inheritance. Which is responsible? DNA. You look like your mother or father or grandfather characters also know due to the presence of DNA molecules. Okay. Okay. The DNA molecules, the nucleic acids also exist in two types. What are they? DNA, and another one is called as RNA. What is by DNA? Deoxyribonucleic acid is called as DNA. RNA means ribonucleic acid. What are the difference between these? Deoxyribo. Deoxy means one oxygen molecule is removed from the ribo. It is a DNA. Suppose ribose molecules, you are adding oxygen molecules. What happened? That is DNA. See here. DNA has a double standard structures. This was discovered by name of the scientist Watson and Crick. Okay. It is a polynucleotide whose monomer unit are deoxyribonucleotide. Just now we studied polynucleotide. Okay. It is also called as deoxyribonucleotide, simply called as DNA. Okay. See that one. The length of the DNA is determined by the number of nucleotide. Take an example. Lower animal is there. For example, house fly or honeybee or any other fruit fly, whatever it may be. Take an example of monkey and chimpanzee or cow or buffalo. These are all higher animals that are all, what do you know, lower animals. Which one it has a high nucleotide, number of nucleotide, which one is higher? Anybody else? Dhanalakshmi?
Sir, higher organisms.
Yeah, very good. Length of the DNA molecule is determined by the number of nucleotide. The number of nucleotide also determine the character of the animals or plant. Don't forget. Length. Suppose 2 meter length is there. It is denoted as human being. Suppose 1 meter length is there. It is also depends upon their number of nucleotide. Okay, and also it indicate the lower animals. Okay, it also depends upon the chromosomal also. Already we studied. We are having 46 chromosomes. The chimpanzee has 48 chromosomes. And housefly and other lower animals, it has 14 chromosomes. Groundnut, it has 40 chromosomes. Onion, 16. Likewise, each one it has a specific DNA length and also nucleotide. Okay. So DNA is a double standard structure, isn't it? But RNA, on the other hand, has a single standard structure. Don't forget, my dear. DNA is a double standard structure and RNA is also a single standard structure. Not only the DNA is a double standard and also exists in different form. What are they, double standard, circular DNA, linear DNA. Okay, these are the types. But RNA, it contains only one strand only, but the structure will be 3 types. Okay, according to their structure. mRNA, tRNA, rRNA. But in this case of DNA, they will not different kinds compared to RNA molecule. RNA, it contains 3 forms, but DNA, it contains only one, but different shape is the double standard, single standard, and also circular standard. For example, in bacteria, okay, the extra chromosomal body is also present in it. That extra chromosomal body is called as, what is that? Extra chromosomal body present inside the bacteria. Take an example of E. coli. The name is called as, what is that? What is that, my dear? That is called as plasmids. Extra chromosomal body. Don't forget, my dear.
Plasmids, sir.
Yes, it is very, very essential for the, what is it, genetic engineering process. Okay, say that one. So it is also a polymer whose monomers unit are ribonucleotide. RNA is also ribonucleotide. DNA is also deoxyribonucleotide. See that one. See, a nucleotide has 3 components. Note it down, my dear. This is very, very important. Already Ajay told. Once again, repeat for you. Don't forget, my dear. The nucleotide, tide, T. T means 3 components. What are the 3 components present in it? First one is pentose sugar. Pentose sugar. That is also deoxyribose. Okay. And also that is called as ribose sugar. See here. The ribose sugar. First one. What is my nucleotide? Nucleotide consists of 3 components. Is the pentose sugar, nitrogen base, phosphate. The pentose sugar, suppose if it is a DNA, deoxyribose. Okay. Suppose if it is RNA, ribose sugars. What about the nitrogen bases? Already but they are majorly divided into 2 types according to their structure. We will see one by one. Okay. Heterocyclic nature, monocyclic nature. Purine and pyrimidine. Purine, it has a heterocyclic nature. 2 ring structure is there. Okay. But pyrimidine, only 1 ring structure is there. Already about the phosphate. These are the 3 components present in it. That is called as nucleotide. Suppose absence of phosphoric acid, only the pentose sugar as well as the nitrogen only present in it. That is called as nucleoside. Okay. Here, one by one, we will discuss. First one is pentose sugar. First component name is called as pentose sugar. So already we discussed pentose sugar, otherwise called as monosaccharide. It is one of the monosaccharide with 5 carbon atoms or sugar molecules. Monosaccharide means sugar molecule. How many carbon atom present in? 5 carbon atom. That's why the name has given as a pentose sugar. Pentose. Pento means 5. Hexose means 6. Heptose means 7. Octo means 8. Erythrose means 4. Xylose means 7. Likewise, different carbon atom is also there. Okay. Okay. See here. The ribose sugar. The ribose sugar, if it is present, means that molecule is called as RNA. The first letter R. Ribose nucleic acid. Because the ribose is named by the ribose sugar. DNA, deoxyribose. Sugar molecules, deoxyribose. That's why deoxyribose sugars. You know the difference between DNA and RNA. DNA means the sugar molecule, deoxyribose sugar. RNA means ribose sugars. You have to remove the oxygen molecules from the ribose. It may convert to DNA. DNA to RNA. This is a circular. RNA, when, what will happen, you know? Removal of the oxygen molecules, it converted into RNA into DNA. Addition of oxygen molecules, what is that? RNA. Okay, see this one. So next one, they are as follows. See, this is the sugar molecules. Ribose, deoxyribose. Both are, what is the 5 carbon sugar? See that this is the carbon atom 1, carbon atom 2, carbon atom 3, carbon atom 4, and also this is the 5th one. Okay. So what is the difference between these two? You know, see the second position. Second position connected by H bond with OH, hydroxyl ion. See this one, hydrogen atoms combine with the hydrogen atom because the oxygen molecule is removed. That's why it is called as deoxyribose. See the second position, the oxygen molecule is present in the ribose. Here, the oxygen molecules removed or absent. That's why it is called as deoxyribose. Okay, so both are same. Deoxyribose is present in it, that is deoxyribose nucleic acid. Suppose ribose molecules present in it, ribose sugar molecule present in it, that is called as ribose nucleic acid, otherwise called as RNA. This is DNA. Okay, you know the difference between these two, my dear? Ajay?
Yes, sir.
Okay, are you clear now in this part?
Yes, sir.
Okay, good. What about Dhanalakshmi?
Yes, sir.
Okay, go ahead. Next one. So, sugar molecule finished. Second major component, nitrogen bases. The nitrogen containing component with properties of a base, 2 types. Already you know. There are 2 types. Is there? One is called as purine, another one is called as pyrimidine. Purine and pyrimidine. Purines first. The purine is a heterocyclic aromatic organic component. Aromatic means it's no variable, but ring structure is there. And also 9-membered ring structure. No, don't forget, my dear. This is very, very important. Heterocyclic and aromatic organic component. Addition with this 9-membered ring structure is also there. Example is adenine and guanine. Okay, adenine and guanine. So purine, heterocyclic, 9-membered ring structures. But the example is adenine and guanine. See the structures. Adenine and guanine. See the structure, you can easily identify. These are the 2 ring structures. See that hexagonal shape. 6 carbon atom, 7, 8, 9. So this is the 6 carbon atom, aromatic ring structure, 7, 8, and 9. Okay. Adenine and guanine. What are the changes here? Just imagine. See, this portion is also changes. This is the, see that this one. H is not here, H is not here, H is present here. What about this area? The oxygen replaced by NH2. Okay, this one. The NH2 is replaced by which, you know, this area. See here, the NH2 is replaced here. Okay. Similar to this structure. These structures are similar. So these are the similar structure. Only the hexagonal structures. Hexagonal means the aromatic structure will be different. What is that? This N gets replaced by here. Here the occupation of the oxygens. So adenine converted to guanine. These are the structures. Sometimes they may ask the questions. These two are molecular weight is very high compared to the other components. This is very, very important, my dear. Adenine and guanine is also high molecular weight, whether both DNA and RNA. RNA, there is a substitutions. I will tell you. Okay, see that one. And pyrimidine. Pyrimidine is a heterocyclic aromatic organic compound, but 6-membered ring. There are 9-membered ring, but in this category, 6-membered ring is there. Example, cytosine, uracil, and thiamine. See that second letter, pyrimidine, second letter, which is what is this? Dhanalakshmi. Second letter.
Sir,
Pyrimidine, second letter, what is that? Second letter, spelling. Pyrimidine, the second letter I am asking. Bad. What about Ajay? I'm showing, isn't it?
Yes, sir. PYRIMIDINE.
So second letter only I asked. Why?
why, sir?
Okay.
Why,
So
sir?
the Y, yeah, okay, okay. Y is present here, the second letter, isn't it? Cyto. Pyrimidine, there is a second letter, Y. The Y is also present in it. Cytosine, the second letter. What about the thiamine? There is a third letter.
Third letter, sir.
Yes. How will you differentiate between purine and pyrimidine? That's why I'm asking. The Y is the second letter. You can easily identify Y. Okay, Y, which one it carries? So they are all are considered the is known as pyrimidine. But uracil is there. What is that? Option. The uracil mostly found in the where? In the RNA molecules only, not in the DNA molecule. Instead of thiamine, uracil only present in it. Okay, see that one. So you can easily identify. Okay, and the other one is called as chemical name. Sometimes they ask this. This is noted down, my dear. Both of you, please note it down, my dear student. This is very, very, very important. So many times they were asked. Adenine. The chemical name is 6-aminopurine. Okay. Guanine. 2-amino-6-oxypurine. What is the meaning of 6-aminopurine? Because the sixth component, which one is there? Amino group is
Amino group, sir.
very good. What about guanine? The second amino 6-oxypurine. So second carbon atom occupied by the amino and the sixth carbon atom occupied by oxygen. That's why 2-amino-6-oxypurine. Cytosine 4-amino-2-oxypyrimidine. And thiamine 2,4-dioxy. Dioxy means 2 oxygen molecules occupied. And 5-methyl pyrimidines. See here, 5-methyl. Methyl means you know where CH3 group occupy in the 5th carbon atom. This is very, very important, my dear students. Don't forget. Please note it down. Don't forget. Adenine, guanine, cytosine, thiamine, and uracil also there. Just to find out the chemical name of the uracil. This is your homework. Okay. But from these 5 components, so thiamine and also uracil, which one is the lower molecular weight, my dear? Which one is the lower molecular weight? Anybody else? Ajay and Dhanalakshmi. Adenine, guanine, cytosine, thiamine, and uracil also there. Which one is the lower molecular weight?
So is it uracil, sir?
Very good. Exactly correct. Okay. So the uracil is the lower molecular weight. But at the same time, I ask one more question. Both DNA and RNA, which nitrogen base is common? Which nitrogen base is commonly present in both? Any idea? Dhanalakshmi?
Sir, thiamine.
No. I ask it, what is that, both Both nucleic acid, which nitrogen base is commonly present? You don't know. What about Ajay? Yeah, you know the answer. You know the answer, Ajay.
Sir, I'm not sure, sir.
Oh, okay, okay. You will try. You will try. The wrong or right, we'll correct it. No need to worry. No need to hesitate. I already told you. These are all very, very important topic. That's why. Please. Okay. I will explain, isn't it? See that next one, you can easily identify. So this is the cytosine and thymine and uracils. These are all concerned that it is known as, it is, pyrimidine structure. This is also hexagon. This is also there. So cytosine, thymine, and uracil. See that lower part, all are same in nature, isn't it? Here the N, here the NH, here the NH is there. Whatever this part. See that NH2 amino group is there. This is also oxygen. This is also an oxygen group. So the cytosine, thymine, uracil. Just to practice it, my dear students, the structure of the purine and pyrimidine. It's very, very important things. Okay. See here. So this is very, very important. See, my dear, nucleic acid, two things is there. DNA and RNA. Okay. DNA is nothing but deoxyribonucleic acid. RNA is nothing but the ribonucleic acid. Deoxyribose, ribose. Purine molecules already in our blood. Adenine, guanine, thymine, and cytosine. This is the DNA molecule component. So RNA molecule component same as it is adenine, but here what happened, What happened, uracil. The instead of thymine, the RNA molecules replaced by uracil. So the thymine is not present in the RNA. Okay. But RNA, guanine and cytosine. Both, which one is common? See that? Guanine and cytosine present in the RNA molecules. Likewise, and also DNA also. Yes. Sometimes they ask the question, these three mostly found in the DNA as well as the RNA. But common they asked, isn't it? So adenine always combined with the molecules of thymine, DNA. But here adenine is also combined with the molecules of uracil. So the possibility is different component is there, joining component. But common is there, guanine always combined with the molecules of cytosine with the 3 bond. And here also the guanine always combined with the molecules of cytosine with the 3 bond. So RNA as well as the DNA, these component will be common. Don't forget. Guanine combined with the molecules of cytosine, one purine molecules combined with the another pyrimidine with the help of the triple bond. So these two are common. So guanine and cytosine mostly common in the both nucleic acid, DNA as well as RNA, because adenine, thymine, okay, adenine is also combined with the molecules of thymine, but in the case of RNA, what happened? Adenine combined with the molecules of uracil, so we cannot able to take in your counting. Okay, so these are the two things is there. So guanine, cytosine. Here also guanine, cytosine. So these are continuously come to the generation to generations. But this is changes. These are the changes between the purine and pyrimidine. So it will not come to the counting. Okay. Okay. See that purine and pyrimidines. Adenine, guanine. Pyrimidines, the thymine, cytosine, uracil. Just to practice it, my dear students, where there is a changes is there, how it will be. Okay. Sometimes they may ask the question, which carbon atom? 9 carbon atom. This one is 6 carbon atom. Heterocyclic. Okay. Lower molecular weight. Only present in the DNA only. Only present in the DNA only. See here, DNA only, uracil. Okay. Thymine is, these are all are what is a high molecular component compared to uracil molecules, because uracil only present in the RNA only, not in the DNA. So this is the higher DNA molecule is a high molecular weight compared to the RNA molecule. Okay. Okay. So and third one is called as phosphate. This one we will finish it up to this level. We will see the remaining on the next class. Phosphate group. Third one is phosphate. First we studied sugar molecule. deoxyribose sugar, ribose sugars. Nitrogen base, purine and pyrimidine. Okay. So last one is called as phosphate group. Inorganic salt of phosphorus forms backbone of polynucleotide chain along with the sugar molecule. The phosphate molecules always joined with the which one the sugar molecule, whether it may be ribose or deoxyribose. Suppose if it's DNA, the sugar molecule and the phosphate molecules connect with the joins. That is called as sulfide bond. So this is a structure. See that nucleotide structure is there. See, this is a nitrogen base. Okay, nitrogen base is there, center, followed by the sugar molecule is there, and also the phosphate. The phosphate molecule connected with the what is that bond between the fourth carbon atom. This one is also fifth carbon atom. Fourth carbon atom is here. Fifth carbon atom. Fifth carbon atom united with the phosphate group. Okay. Likewise, another strand is also there. Opposite strand, complementary strand is also there. It is also connected with the another phosphate group. That's why disulfhydryl phosphate. Disulfide ester. Okay. See that these are the things is there. So we'll see this part in the next classes. Today, what we'll have discussed, just to summarize all those things, we will pass on to the next classes. See here. So what is meant by DNA? Okay. So what is meant by genetic material? Haveli Kloof. Who discovered DNA as a nuclein? Who discovered RNA? What is Chargaff rules? How many types of bond formation in DNA molecule? These are the things we are going to study in these lessons. And what is this higher molecular weight to and lower molecular weight in order? Okay. Genome, chromosome, gene, and nucleotide. Before that one, there is a DNA, nucleotide, DNA, and also alleles, nucleotides. Okay. So this is the correct way of order. Okay. Don't forget, my dear students. So this is the things. Already discussed what is by genome, what is mean by genome, what is by chromosome, what is by gene, what is by nucleotide. Okay. So here, so what is by DNA? Whether it's acid or base, both the characters present, which means both acidic character as well as the base also present in the single components. That's why it is called as amplification nature. And discovery of a gene, this historical aspects already in our about 1868, John Frederick Melcher, okay, discovered from the pus cells. He gave a name, is called as what is a nuclein. It composed of what is that, DNA and protein molecules. Okay, so that one. And next, Levon, P. Arnon Levon, discovered the nucleotide or named as a nucleotide. 1953 is very, very important. Year is very, very important. Double helical structure DNA, Watson and Frederick, Francis Crick. Based on the X-ray diffraction of the, which one Maurits Wilkes and Rosalind Franklin. Hallmark, what is the main hallmark of this one? Base pairing. Base pairing is also very, very important. It is also observed by the Erwin Chargaff. Erwin Chargaff rules, what is that? One adenine molecules combine with the another, and one purine molecule combine with the another pyrimidine. Okay, that's why. So adenine and thymine, guanine and cytosines. Okay, so both are equal in nature. Adenine is also equal to the thymine. Guanine is also equal to the cytosines. Adenine combined with the molecules of thymine with the help of double bond. Guanine with the triple bond. Don't forget, my dear students, both are equal in nature and also one. Nucleic acid, DNA and RNA. Already we studied. Polynucleotide, otherwise called as what is that? Nucleotide. Length of the DNA molecule determined by the number of the nucleotide. RNA is also single standard. DNA is a double standard. It's nothing but the polymers, more than one molecule. This is also very, very important. Sometimes they may ask the question, which one is polymers? Polymers of monomer unit are called as nucleotide. Same components repeated again and again and again. That's why it is called as polymers whose monomer units are ribonucleotide. Okay. So this is the, what is the three component nucleotide? What is nucleoside? Nucleotide means Three components: pentose sugar, nitrogen base, phosphate. What are the pentose sugar? Suppose if it is RNA, ribose sugar is present. Suppose if it is a DNA, deoxyribose is present in it. Okay, so these are the structures. Which position? The second carbon atom, the oxygen is left. So deoxyribose. Presence of oxygen is called as what is a ribose sugar. Okay, so remaining things we will see the next classes, my dear student. Thank you for the participation. Please don't forget this part. Okay, don't left it. And from these two lessons, the last one as well as this one, don't forget 12 question must be there. 12 question, not less than 8. Okay, so don't left. Please follow it. Take a notes, my dear student. This is very, very important. Okay, thank you so much for my participation.
Thank you, sir.
Thank you, Bob. Thank you, my dear.
Thank you,
Ajay.
sir.
Thank you, sir.
Thank you, ma'am. Thank you, ma'am. Thank you.
Thank you, sir.
Thank you, sir.
Right, Kana. Bye.